Hello, my name is Jacob Schreiber, and I'm a postdoc working with Bill Noble at the University of Washington. Today, I'm going to be giving a presentation entitled Zeroshot Imputations Across Species are Enabled Through Joint Modeling of Human and Mouse Epigenomics. In the past at ISMB, I've introduced a model called Avocado that is capable of imputing genome-wide functional experiments. Here we have two examples of the experimental data and imputations for the assay H3K27 acetylation and the protein CTCF in HepG2. You can see that the imputations match the experimental signal fairly well. This work was motivated because we have only performed a small number of the potential experiments that we could in humans, or ever will be able to, because of the cost of performing one of these experiments. The goal was that if we can build a model that takes in all of the data that has been collected, we can impute the tens of thousands of experiments that have not yet been performed. Ultimately, this effort was fairly successful, and so we were eager to try to apply it to species outside of humans. However, upon applying it to mouse, we immediately ran into some problems. The first problem is just that there's far less data available in mouse than there are in humans. On the ENCODE project portal, there's almost 10,000 experiments mapped to the human reference genome HG38. However, there are only around 1,700 experiments mapped to the mouse reference genome MM10. This lack of training data means that the model will have less available when trying to make imputations for the missing experiments. But perhaps more importantly, the experimental matrices themselves are somewhat limited. For human data, there are over 1,000 biosamples that have at least one experiment performed in them, and there are over 1,300 assays that have been performed at least once. In contrast, in mouse, there are only 114 biosamples where at least one experiment has been performed, and only 71 assays that have been performed at least once. This is a big problem for modern imputation methods because they are restricted to the set of biosamples and assays that have at least some experimental data for them. This limitation in mouse means that imputation methods as they currently stand will be limited in the number of biological phenomena that they can even make imputations for. So this motivated the research question for this project, which is, can we leverage the large amount of available human data when making imputations in other species? This is kind of broken down into two sub-problems that we investigated separately. The first is, can we improve imputations in this traditional fill-in-the-matrix imputation task? Basically, when leveraging this human data, can we just do better at the standard task of making imputations? The second is, can we make more comprehensive imputations? In this case, what we want to do is we want to make imputations in mouse for assays that have only been performed in human. This is an example of a zero-shot problem, which is where you're trying to make predictions in a setting where you don't have any available training data. In this case, what we're trying to do is we're trying to make predictions for assays that have only ever been performed in human. In its original formulation, Avocado is a deep tensor factorization method that is applied to a compendium of data from a single species. It is a tensor factorization approach because it organizes this compendium into a 3D tensor, where the axes correspond to the assays, the cell types or biosamples, and the genomic positions, because each one of these experiments yield genome-wide measurements. It then learns low-dimensional linked representations for each of these three orthogonal axes. Avocado is called deep because rather than using a dot product in order to combine these low-dimensional representations into a prediction, it uses a neural network. The weights of the neural network and the latent representations themselves are then trained jointly on the task of imputing or predicting values within the tensor. In this manner, you can then substitute any combination of genome position factors, assay factors, and biosample factors together in order to impute any of the experiments within the tensor. However, we had to make a few modifications in order to get Avocado to work in a multi-species setting. Specifically, we had to join data together from these species. So what we did is we used genome alignment in order to map data from human experiments into positions within this tensor of mouse data. What you can see here in the schematic is that basically you'll have blocks of data available for mouse positions that align to the human genome, but you won't have any data available for the positions that don't align to the human genome. After mapping this data over from humans using the genome alignments, the resulting model has a biosample axis that contains the union of mouse and human biosamples. It has an assay axis that contains the union of assays performed in mouse and assays performed in human, but the genome position and the neural network components are the same as before. Because this new data tensor is going to have far more human experiments in it than mouse experiments, we then had to change our sampling strategy in order to equally sample from both species. This ensures that the human data doesn't overwhelm the mouse data that we have available. We then had to use a few other tricks in order to get the best performance of our model, but I'm going to skip over those for now. So we then wanted to see how well this new approach would work when making imputations in the traditional imputation setting. We compared against a few baselines. The first baseline was the average activity. This is the per position average signal for each assay across the training set. Essentially, it's basically just saying how much of this signal is similar from one biosample to the next. And usually it's a pretty strong baseline because a lot of signal is similar across biosamples. It also has the convenient property that generally when you outperform the average activity baseline, it means that you're making biosample-specific predictions. We then compared against an Avocado model that was trained using only the mouse data. And so this is just the performance that we'd have before using human data at all. We also trained another model that was trained using human data but didn't use any of the alignment information. And then finally, we had our model, which incorporated all of this information into a single model. What we observed is that the more information that the model had access to, the better the predictions that it made. We can see that the overall error decreased from 0.08 to 0.057 when used between the average activity in our model. When we broke down this performance by type of biological activity, we observed that we saw the largest absolute gain in performance when looking at histone modification experiments. However, we observed the largest proportional gain in accuracy when looking at transcription experiments. This is important because transcription is a complex phenomena, and this shows that by incorporating human data, we're able to learn about this much better. This is also a finding that David Kelly saw when he trained his Basenji model using both human and mouse data. So it's reassuring that seeing this large proportional gain when modeling human and mouse data together jointly is something that is being seen in multiple contexts. We then wanted to evaluate where the improvement was coming from, and in doing this, we identified two distinct types of loci, sets of loci with distinct characteristics. On the left-hand side here, we had one set of loci where the mouse-only model was improving upon the average activity baseline, and our proposed method continued to improve upon that. In these loci, mostly what was happening is that our method was getting the signal value more correct than the mouse-only model. And so while this improvement is important, it also represents kind of a marginal improvement in that you're just continuing to do slightly better at the positions where we are outperforming the average activity baseline already. The more interesting set of loci were the second ones on the right-hand side. At these positions, the mouse-only model had kind of overfit to the average activity baseline and were making the same incorrect predictions. You can see here that's just not predicting anything for the total RNA-seq assay here. However, our methodology allowed it to kind of break away from this average activity overfitting and make predictions in a biosample-specific manner. So the differences that are occurring at these loci are much more significant than the ones that we're seeing on the left-hand side, and truly represent the model learning something that it couldn't have learned before. So we then wanted to look at the Zeroshot setting more. And in this setting, we focused on protein binding experiments because these experiments are far sparser in both human and mouse, but also because they play a crucial role in understanding gene regulation. And so what we did in this setting is that we split the assays themselves, as opposed to the experiments, into 3 folds. And what we did is that we would train a model using all of the experiments from 2 folds of protein binding assays, as well as all of the other mouse data that wasn't related to protein binding, and all of the human data. This is similar to what we did in the previous cross-validation setting, except that we're splitting the assays themselves into folds rather than the experiments. And so then we would evaluate how well we can make predictions for all experiments in the held-out fold of protein binding experiments. And so this really is zero shot, because the model is going to have access to no mouse protein binding data for that fold of assays. So there wasn't really a good baseline for us to compare against, because the average activity and the mouse-only model relied on you having data available for each assay and for each biosample. However, in the absence of a good baseline for us to compare against, we just continued using the average activity as our baseline. So this is going to represent a very strong baseline, because it explicitly has access to information that our model is not going to have access to, specifically signal from this type of assay just in a different biosample. So we first we calculate the average activity for each one of these assays. Then, like before, we trained a model that used the human data but did not use the alignment information, and we saw an improvement over the average activity. And then we trained a model using both the human data and the alignment information, and we saw another significant improvement. So what we're seeing here is that even though the average activity baseline is strong, that our method is able to significantly outperform it. We're able to reduce the mean squared error by a lot. This means that we're able to make biosample-specific protein-binding imputations even in the zero-shot setting. We were then kind of interested in seeing how poorly are these imputations going to be? How poor are these imputations going to be when you try to make predictions for a protein that's very dissimilar from any of the ones that you already have data for? And so we trained models in a different setting, which is that rather than having access to some but not all protein binding assays, in this setting you have access to no protein binding assays in mouse at all. And so this model was trained only using non-protein binding data in mouse, and only sees the protein binding data in human to understand what protein binding means. And so we trained human data and a human data plus genome alignment model. And what we saw is that while the model that only used human data performed fairly poorly, that the model that used human data and the genome alignments performed similarly to the average activity. This is pretty surprising to us, because what this means is that even when you make predictions for a protein that's very dissimilar from all of the ones in the training set, that you get performance similar to having seen that assay in a different biosample, which is what the average activity is. This is really strong performance, and so we were really happy when we saw this. So we then broke performance down by assay, and what we observed is that for around 1/3 of protein binding assays, we saw an improvement in the mean squared error, a decrease in the mean squared error of around 50%, which is really large. For 2/3 of assays, we saw a decrease in the mean squared error of at least 20%, which again is a pretty large decrease. Over all assays, we saw an average decrease in the mean squared error of 33.6%. However, not all assays showed an improvement. There were 2 assays that didn't. The first was USF1, where the decrease in performance was basically negligible, and the second one is CTCF, which, as we all know, has no biological meaning at all. But in this case, CTCF is, it's not really surprising that the average activity was strong for CTCF, that CTCF has very similar binding across biosamples. It binds very tightly to its motif, and CTCF is the most assayed biosample, is the most performed assay in mouse. And so the signal that you're going to get is going to have very little noise. We then wanted to, like before, investigate what the source of the improvement in our imputations were. And so what we did is we wanted to see whether or not the protein binding was occurring in a more concentrated manner in chromatin accessible sites than the average activity. So what we did is we phrased this as a classification task. We wanted to see whether or not the protein binding imputations were able to recover DNase peaks. So we binarized the experimental DNase peaks, and we saw whether or not the corresponding imputations of protein binding in those assays in those biosamples was able to recover those peaks. What you can see here is that Avocado is doing significantly better than the average activity. Each one of the green-colored points is an experiment that had higher recovery of DNase than the average activity, whereas orange is where the average activity outperformed it. Now, this metric could be gamed a little bit, where if the model ended up just predicting protein binding in every DNase accessible site, you get very strong performance, but biologically it would be pretty implausible. So what we did is that we broke this down further and calculated precision and recall. In order to do this, we just binarized the imputations themselves in a similar manner to how we binarize the experimental DNase signal. In this context, the recall is going to be the percentage of DNase peaks that are covered by the imputations, whereas the precision is going to be the percentage of imputed peaks that fall within accessible sites. What we're seeing is that it doesn't look like Avocado's imputations are having a significantly higher recall than the average activity, but that the precision is significantly higher. Phrased another way, what this means is that we're not covering more accessible sites with the imputations. It's just that more of the imputed signal falls within accessible sites. And so the average activity is more of the average activity is falling within chromatin inaccessible sites. So this is really good. It means that we're not just gaming the metric by predicting transcription factor binding at every accessible site, that we're doing it in a biosample specific manner, and that our predictions are falling within accessible sites, which is biologically relevant. So all of the code and the imputations are available on GitHub. You can go and check it out. We have code there available for all of our papers related to this project. And then we have a preprint on bioRxiv that you should feel free to read. We've submitted it to a conference, and so hopefully it gets published soon. I'd like to thank my collaborator Deeply for working with me on this project, and my advisor Bill for giving me money while I worked on it. Thank you.